Hello, everybody. Thank you for coming. So today our speaker is Jay Goldberg, who's going to talk about Datura and the interesting polymorphism in Datura. And right now, he's a postdoc at the University of Arizona in the Department of Ecology and Evolutionary Biology. Before that, he did his undergraduate work at the University of Puget Sound in Washington, and then went on to do a PhD at Indiana University in Indiana. And yeah, so I'll let him share slides and take it away. So thanks, everybody. Thanks, Jay.
Yeah, thank you for having me. Yeah, there we go. Okay. Yeah, thank you for having me, and thank you all for being here. I'm going to be talking about a couple chapters of my dissertation work titled A Sticky Situation. I investigate the factors that are maintaining a trichome dimorphism in Datura wrightii. And so Datura wrightii, it's a solanaceous shrub that I think most of us are probably familiar with. It's native to the Southwest United States and northern Mexico. It loves disturbed soil, so it's really common along roadsides and in human impacted areas. And it's very toxic and drought tolerant, which has allowed it to succeed in the desert environment. And so trichomes are above ground plant hairs. And I'm going to talk a lot about different kinds of trichomes. They have a lot of functions to protect against different abiotic and biotic stressors. I'm going to focus more on the biotic angle in my talk today. And so they reduce the quality of host plants for different herbivores. And they can also, when they're sticky, ensnare and kill small arthropods. This was something that fascinated Darwin. He actually wrote a whole paper about how cool he thought the sundew plant was. And so in Datura wrightii, trichomes are dimorphic. So there is a sticky morph that is covered with glandular trichomes. They exude a sticky exudate full of secondary metabolites. And there are non-glandular plants that feel velvety to the touch, and they're producing exclusively these non-glandular trichomes that lack these secretory structures. Both of these morphs coexist all across California in populations. And so here, this is just an electron micrograph of tomato stem that shows these glandular trichomes with their bulbous heads of secretory cells next to non-glandular trichomes that look just kind of like regular hairs. And so this morph, it's got a single locus inheritance where the sticky morph is dominant, or the sticky allele, excuse me, and the velvety allele is the recessive one. And all plants are actually sticky as seedlings, and around the 5th true leaf, they mature and stop producing glandular trichomes, or the velvety morph does. And so Datura wrightii, because of it's so toxic, it has a relatively small community of herbivores that feed upon it. They're all pretty specialized to deal with toxic alkaloids. First, I think many people are going to be familiar with is the tobacco hornworm, Manduca sexta. There's also the potato beetles, Lemma daturaphylla. It's actually a specialist on different species of Datura. The tobacco suckfly, Tuberculosis mutatus. It specializes on various sticky plants out here in the Arizona deserts. There are flea beetles and a specialized Datura weevil, Trichobaris compacta, that exclusively uses Datura plants. And so I looked at them as adults during my study as the larvae are pith-feeding herbivores, and I didn't feel like going out and completely destroying the plants to find them. And so the two morphs, or rather a plant's trichome phenotype impacts which herbivores are going to feed upon it. So Manduca sexta will feed on both morphs, but the larvae tend to grow smaller and be a bit more sluggish behaviorally when they're feeding on a sticky plant. The same is true of Lemma daturaphylla. the mirids, tuberculoris, they, as I mentioned, specialize on sticky plants. And so you very rarely find them feeding upon velvety plants. But sticky plants can actually be completely infested with hundreds or even thousands of these small insects. The flea beetles are the opposite. They exclusively feed on velvety plants. And the weevils so far don't really seem to mind. They feed on both, and it's a little unclear how the alkaloid or the secondary metabolites being exuded by sticky plants impacts them. So I'm going to talk about two questions that I looked at during my PhD. The first is negative frequency dependent selection responsible for maintaining these dimorphic populations across California? And then to what extent are associational effects between neighboring plants occurring within these dimorphic populations? For both of these, to answer both of these questions, I used a field observational approach. I just revisited populations that were studied previously, counted all the plants present, and noted whether their phenotype was sticky or velvety. This is just showing the diversity in these different populations that I was visiting. So Millerton Lake State Rec Area was an actual natural population on a lakeshore, whereas this was just a roadside lot full of Datura on the side of the road in the Central Valley. And so these are all the populations I visited. They range from just south of the Bay Area all the way down to Orange County. And so things changed over the 20-year period between the previous study done by Dan Hare and my own study. And so I also collected for a sample of plants the number and phenotype of the neighbors, estimated the leaf area that was damaged by all the different herbivores, and noticed whether or not there were actually individuals of these insects present. And so each herbivore leaves a distinctive pattern of damage, which allowed me to figure out exactly who was eating a given plant. Lemma beetles leave these haphazard chew patterns and often avoid veins in the leaves. Flea beetles leave this puncture wound damage. Manduca sexta just completely defoliates and eats leaf and stem and vein all. And the tuberculoris mirids leave these yellowing chlorosis damage. And so, yeah, first I wanted to address whether or not negative frequency dependence selection is responsible. And so this is a selective regime where rare morphs have a fitness advantage, and this in turn drives stability in the polymorphism. So in these examples, right, the fitness of an individual is very high when its phenotype frequency is low, and its fitness is low when its phenotype frequency is high. This in turn can cause oscillations in the phenotype frequency, and neither morph is ever able to fix in a population because its fitness becomes very low when its frequency is very high in a population. And this has been shown previously to act on a trichome dimorphism in a species of Arabidopsis. So this species of Arabidopsis has a hairy morph covered with with non-glandular trichomes, excuse me, and a glabrous morph that lacks trichomes altogether. And they found that reproductive output was higher for the Rare-merph in their mesocosm studies. And so these middle bars are the glabrous morph, and the hairy morph are rare. And you can see that reproductive output in terms of meristems and flowers is higher when either of these morphs is common. And so I hypothesized that I would see a negative correlation between the change in the sticky frequency over time and the literature sticky frequency that was published in 2001. So this is just an example of what I would see. So we would see this larger positive change when the sticky frequency was historically low. And we'd see the opposite. We'd see a negative change when the sticky frequency was historically high. And so this alone wouldn't be enough to rule out other evolutionary forces from acting on these populations. So I assembled a table of predictions that outlined what I see, depending on which evolutionary process is dominant. So the three things that I was looking at are the mean frequency. Oh, there shouldn't be a delta there, but yeah, the mean frequency, the relationship that I mentioned before of the change versus the frequency, and the variance in the frequency. And so drift would cause no net change to the mean. The slope would be 0 instead of that negative relationship that I showed before. And the variance would increase. For migration, we would again have no net change in the mean. We would see that same negative slope that I predicted would be present if negative frequency dependent selection was acting, but we would see a decrease in the variance. Oh, spoiler alert, sorry. Directional selection would change the mean either positively or negatively, depending on the direction of selection. You get either a positive or negative slope, again, depending on which direction the selection is acting in, and variance overall would decrease. And finally, negative frequency dependent selection, we would see no net change to the mean. We would see that negative slope that I described earlier, and we would see no change to the variance. And so what I found was that negative relationship between the change over time and the initial sticking frequency was there, and it was significant, indicating the potential for negative frequency dependent selection to be occurring. This allowed us to rule out drift and directional selection, but as I mentioned, migration will show this same relationship. So I had to do some additional tests to rule out the presence of migration being a dominant force. And so I just looked at the change in the mean and the change in the variance, neither of which changed from the 90s to the 2010s. And also, when I looked at the relationship between the sticky frequency difference between two populations and their geographic distance, I found that there was no relationship. So each point in both of these figures is a pair of populations, and you can see that there is no relationship. So populations that are close together are no more likely to have similar sticky frequencies than populations that are very, very far apart. And so together, these results ruled out migration as a strong force in this system. And we were able to conclude that negative frequency dependent selection is the dominant evolutionary force acting to maintain this dimorphism in California Datura wrightii. But we still wanted to know who's responsible, what agents are out there driving this selection. And so when I looked at the relationship between sticky frequency and the average amount of herbivore damage on plants within a population, I did find two interesting relationships. So in both of these figures, each point is a population's average. And so here we see the average damage from the tuberculosis mirids. And as the sticky frequency increases, you get a higher amount of average damage from this species of herbivore. And the opposite was true for Lemma So as the sticky frequency increased, you get less average damage from this herbivore. And so this strongly suggests that these two herbivores are driving the selection. So when sticky becomes dominant in a population, this herbivore comes in and selects against it. And the same when Velvety, this herbivore, the Lemma beetles will come in and select against it. But this doesn't tell us what's going on within populations to generate this selection. We're just looking at statewide trends. And so I wanted to know what role associational effects are playing in this system. And so associational effects are when you have a neighboring plant impacting the relationship between a focal plant and herbivores. They can be beneficial from the perspective of your focal plant. This is called associational resistance. And so having a neighbor makes you more resistant to an herbivore, or they can be negative from the perspective of your focal plant, where having a neighbor makes you more susceptible and more easily damaged by a given herbivore. And these effects have been shown to occur in both mixed cultures or communities of plants, but you can also get them in polymorphic monocultures, which is the system I was looking at. And so I hypothesized that herbivory will be correlated with more frequency in the immediate neighborhood of a plant, which I defined as within 1 meter. And so I visited populations 3 times to collect data for this study. I went once in 2017 over the summer, and I went twice in 2018, early in the season in the spring, and at the same time that I visited in 2017, later on in the summer. And I had a sample of 25 populations in 2017 and 34 for both visitations in 2018. And so what I found was that velvety plants with sticky neighbors tend to experience associational susceptibility. And so this graph and the next few graphs I'm going to show you are the results of binomial analyses. So I collapsed the data down to ones and zeros. All these data points are individual plants within populations. And you can see they all fall in a 0 where this herbivore did not occur, or a 1 where it did. And the evidence for associational susceptibility is a significant positive trend in this blue line, which represents velvety focal plants. And so on the x-axis, we have the sticky frequency within the immediate neighborhood. And you can see that velvety plants with all sticky neighbors are more likely to be infested with this herbivore than velvety plants with all velvety neighbors. And so this relationship held true for both the probability of individuals occurring and the probability of there being eggs present in both 2017 and 2018. I also found that Tuviocorus notatus, these sticky specialist mirids, were also responsible for exerting associational susceptibility. So in the spring of 2018, I again found this positive slope for velvety focal plants, indicating that when they have lots of sticky neighbors, they're more likely to be attacked by this herbivore than velvety plants with no sticky neighbors. And this relationship was true for both the probability of damage from this herbivore occurring on a focal plant and the probability of those of the species just being found on a focal plant. And so I had a lot of other results that I'm not going to show you the figures for, but I will walk you through them. So first, sticky Datura wrightii, as we expected, were more susceptible to Tuviocorus notatus all across the board. So both the likelihood of them occurring on a plant and the magnitude of herbivory, sticky plants were always more susceptible. Flea beetles were very rare in dimorphic populations. So there were a couple monomorphic populations out near Joshua Tree in the desert where sticky plants were absent that we saw flea beetles, but otherwise they were exceedingly rare in our sample. Velvety Datura wrightii appear to be more susceptible to Lemma even though this herbivore will feed on both, unlike the mirids, it's likely to feed on both. And trichomorph had no effect on Manduca sexta herbivory, which is unsurprising given that it's unlikely that the adults can tell the difference between these two morphs, as flower structure or flower morphology, flower odor, as far as we know, is identical between these two morphs. And so just to summarize all the results that I've presented so far, it appears that there's negative frequency dependent selection acting on these dimorphic Datura populations, and that two herbivore species are responsible: Lemma daturaphylla, which preferentially attacks the velvety morph, and Tuberculosis notatus, which preferentially attacks the sticky morph. And these same two herbivores appeared to be responsible for associational susceptibility against velvety individuals. So there appears to be a spillover effect of Tuberculosis notatus from their preferred sticky morph to neighboring velvety plants. And Lemma daturaphylla appears to have a strong preference for velvety plants, such that in mixed locations, they will seek out and attack velvety plants. But I had another figure that I did not fit in to the talk today that shows that when presented with single individual plants, so with no neighbors at all, they're equally likely to feed on both morphs. But when presented with multiple options, they will feed on their preferred morph. And so before I let you go, I want to just briefly talk a little bit about my current research. So I'm still working with Datura plants, but I'm trying to update my toolkit into the 21st century and use genomics tools to investigate whether or not there's coevolution occurring between Datura wrightii and the Manduca. So Manduca are the main pollinators of this plant, and also one of the most devastating herbivores. A single individual can completely defoliate a plant down to the ground if left to its own devices. And so there's the potential for a coevolutionary relationship to be occurring between these two plants. And what makes it extra cool is that you're getting both an antagonistic relationship and a mutualistic relationship within a single interaction. And so there's actually two species of Manduca that feed on Datura here in Arizona and the American Southwest. There's Manduca quinquemaculata and Manduca sexta. So Manduca quinquemaculata is limited to North and Central America, and its closest relative is actually the Hawaiian hawkmoth. Whereas Manduca sexta has a Pan-American range. You'll find them feeding on tobacco and tomato crops from Canada all the way down to Argentina. And its sister species are two Caribbean hawkmoths. And so just based on this, I have hypothesized that quinquemaculata may actually be co-evolved with Datura, but I think Manduca sexta perhaps might not be, because it appears to have some traits that make it better able to colonize new areas, adapt to new host plants, whereas Manduca quinquemaculata seems to be more specialized for the native plants down here in the Sonoran Desert. So I have two studies in the works. I'm doing some phylogenomics, comparing my de novo genome assemblies of Datura wrightii and Manduca quinq to existing Solanaceous and Sphinxid genomes. And then I'm hoping to do a population genomic studies of mutualism by resequencing both plants and moths from across the range of Datura wrightii. So, I mean, what can we learn by comparing new genomes to existing ones? We're a little limited on the moth side of the equation. There's tons and tons of Solanaceous genomes out there, as I'm sure most of us are aware. But luckily, there's a recently sequenced hawkmoth from Europe that has no natural interactions with Datura wrightii over its evolutionary history, or any Datura species, as Datura is a New World genus. So this is an interesting comparison that I hope yields something interesting that I can show you at some point in the future. And then, so for the pop gen, I'm gonna go around. This is approximately the range of Datura wrightii, at least in the United States. I'm not entirely sure what its range in Mexico is like, but if anyone knows more, I would love to chat about it. And so I want to test Jeremy Yoder's models of mutualism dynamics. So he has 4 non-mutually exclusive hypotheses as to how plant pollinator mutualisms can arise. And I hope to, with this data set, test those hypotheses and find out which models fit the Duca-Datura relationship. Yeah. And with that, I would like to acknowledge everyone who helped me with these projects, my PhD advisor, Linda Delph, and my committee, Dan Hare for providing me his data, IU, the biology department, my funding from the NSF, and my current project with Judy Bronstein, Luciano Matskin, and Mike Barker, all helping me out a lot, and additional funding from the NSF that's made this project possible. And with that, I'm happy to take any questions, and please, if anything pops into your head after this, please feel free to email me. Yeah, thank you.
Awesome. Thanks, Jay. Yeah, that was super interesting, and I'm sure there'll be questions. If people have questions, just type "I have a question" or "question" in the chat, or if you don't want to turn on your audio, you can type the whole question and I can read it for you. It looks like we do have a question from Federico, so go ahead.
Yeah, hi. Nice talk. I wanted to ask you a question about the genetics and metabol. Do you hear me? Yes, I think so. Yeah. The genetic and metabolomic base of these 2 morphs. So is there something known? Because they occur in the same population, so they can cross. Is it like a on-off trait, or are there intermediate things? And what is now the chemical differences?
So we don't actually know. So I'm the first person to try and sequence and actually look now at the species. But I mean, because it's I mean, it follows single locus Mendelian rules. We think there's a master switch somewhere. So they've identified some trichome master switches in the Arabidopsis genome. So those are probably going to be the first loci that I start looking at when I can.
So the differences are probably due to trichome associated metabolites or something like that, plus the physical effect of the trichomes in terms of the herbivory. Or do you think there are volatiles too that might affect the interaction with the insects?
Oh, so that I have. So as far as we know, the plants themselves and their induced volatile profiles are the same between the 2 morphs. But the glandular trichomes can actually, there's metabolites in them that alter the herbivore odors. And so that's something that I'm looking at. I think those odors might be attracting lizards, actually. But yeah, as far as we know, the plants themselves smell identical.
Okay, thank you.
Yeah.
Now I have to know, what kind of lizards do you think that they're attracting?
So they're called whiptails. The genus is, Spitoscelis. Yeah, they're super olfactory oriented. And they respond. So I've gotten them to try to eat cotton swabs when I put different herbivore odors on them. Yeah,
that's pretty cool. Any other questions? Yeah. Sorry, I didn't mean to jump in front of you there. Richard, you want to go ahead?
Thanks, Jay. That was really very cool project and results. But I'm curious about the T. notatus that is the specialist on the ones that have the sticky glands.
Mm-hmm.
Is there any evidence that the sticky glands themselves are a resource for those insects as a food source or a defense mechanism or anything?
So yeah, there is some potential for it to be like a predator escape. So some predators are very deterred by the sticky trichomes. Others actually seek out sticky plants. So it's a little unclear from that angle. And as far as a food source, so they're a piercing sucking insect, and so they're just eating mesophyll. And so which is actually really cool because they totally avoid all the like sand and debris that stick to the plants that might deter like chewing type insects.
Neat.
Looks like we've got a question. I can read this one. How much variation is there within the glandular plants in trichome number? And do you see any herbivory patterns in glandular plants with high trichome densities versus low trichome densities?
Oh yeah, so there's very little variation in trichome density. So I didn't actually look at that explicitly, but Dan Hare did a bunch of preliminary work showing that it's very, very low.
Cool.
Or the variation is low, yeah. So
Stacy has a question.
Yeah, I was just excited at the thought there's going to be a Datura genome, and I seem to feel like I saw some other resources coming out, and so I was just wondering. I mean, it'd be really cool to have a few Daturas, and I'm trying to remember from the phylogeny, like I should know. Is wrightii, because I'm guessing the other one that would get sequenced is the stramonium. Is stramonium and wrightii, are they closely related? Like if we got those two genomes, could we shoot?
stramonium's already been sequenced, so
Okay.
that was published in January, I think, the stramonium genome. But yeah, wrightii is not. It's like on the other side of the Datura tree, pretty much.
So are these the only ones coming out, or do you know of other people working on other Datura genomes?
Not that I'm aware of. So
Well,
yeah.
having two is going to be super. Now somebody just needs to sequence a Bergmansia genome. I wouldn't be surprised if Federico is going to do that.
That would be wonderful.
Anyway, thanks for the talk. It was super interesting.
Oh yeah, no problem. Thank you.
All right. Well, I'm not seeing any additional questions pop up. Last chance, ask questions. All right. Great. Well, thanks again, Jay. That was cool, and I'm glad you came to give us this talk.
Yeah, thanks for having me.
All right. Well, so sorry, I'm very unprepared today. Next week, let me take a quick look, so I can ensure I'm telling everyone correctly. So next week, October 15th, we'll have Jill Miller from Amherst College, who's going to talk about within species polymorphism and sexual strategy, mating system, and ploidy level in species of Lycium. So that should be also interesting, and hopefully we'll see you all next week. And thanks again, everybody.